So Manfred is going to tell us about, we are very pleased to have Manfred Barton from UC Santa Cruz. And he's going to tell us about the blessing and curse of an algorithm.
Right, the multiplicative update. I've worked on this for a very long time, and this topic makes me tick in various ways. As a human citizen on the Earth, I think about globalization. I also have a farming background. I garden a lot. I do a lot of different things, and this ties it all together, and hopefully it will be obvious. So what is a multiplicative update? It's an algorithm that maintains weight vectors, and the weights are multiplied by non-negative factors. And this occurs, you're forced into this regime when you have concentrations. And concentrations cannot go negative. This is my sense of it. So nature, because it has concentrations of genes, it has number of species that survive of a particular kind. You're already immediately forced into this regime. In machine learning, we have other methods. We have additive updates where you do gradient descent, you ski, but then you have to have negative weights, and that's fundamentally different. So this is the multiplicative. It's motivated by relative entropy as a regularizer. Okay, here's the simple, typical machine learning setting. I'm going doing very simple machine learning multiplicative updates. They are kind of asexual, totally boring, but I want to get a point across. And then I add always a little bit of a trick to improve them, and I show that that trick is necessary and what it does. So typically we have a set of experts that do something on a trial-by-trial basis, and SI is the share of a particular expert. I used some biology notation because I teach classes with biologists and economists on evolutionary computation and stuff like that. So SI is sort of the weight of the ith expert, or the share of the ith expert, and then the multiplicative update is something like new weight is old weight times a factor, non-negative factor. That's very important. It's e to the minus eta loss. Loss could be a fitness rate, and this is the fitness. In biology, this is usually called big WI. And then you normalize. And I claim that Bayes is a special case of this. Bayes is the fundamental thing for updating conditional probabilities. Bayes rule. If you put a log likelihood up here, then this becomes posterior is prior times data likelihood over normalization. I will bring that obvious in a moment, and I'm going to show how to implement Bayes at a very large scale. So the weights in nature are concentrations, shares of genes in a population, and they update it multiplicatively. That's the connection to these machine learning algorithms. We didn't know this connection at all when we started doing multiplicative updates. First person was in my community was Nick Littlestone, and then we did lots of work on this. I've been working on this ever since, but the connections to biology only in the last 10, 15 years. Okay, good. So the blessing of the multiplicative update, as I will bring out, is its speed. It does multiply by factors, so you get sort of this exponential growth. Okay, and that's a problem. It's good and it's bad. Okay? It's bad because you have a loss of diversity. If you run this algorithm for a long time, many iterations, one guy wins, and you have die out. And that is why multiplicative updates are good, and that's why they're bad. And if you don't have a mechanism for fighting the curse, you will be in trouble, and I will bring that out. Can I show you how we do it in machine learning? And then I made more and more connections to biology, and it's kind of mind-bogglingly interesting, I think. Okay, so we'll give 4 machine learning methods for which we can prove things. I don't know anything about proofs here, but we can prove things. They all sort of information theoretically optimal. And then I discuss related ones in nature. Okay, next I'm going to implement the Bayes in the tube. For that, I do a totally different trivial multiplicative update, and that is in vitro selection. Done in the 1960s, Orff started it. So you have a soup of random RNA strands in this tube, right? Here's my cup. I have props. Okay, so, and you tip in a sheet of RNA, a sheet of protein, and what you want to do is you want to select for a strand that attaches to the protein. This is true, this is important for medicine because you have to get the agent to the place where you want it, okay? So you tip, pull out, wash off the RNA that's stuck, and then multiply washed off RNA back to the original amount. Because you have to amplify. Because when you only a small amount attaches, so now you have to amplify what attached, this is the good stuff, to the unit amount. So you have still the same amount of RNA you had before. Okay? So this cannot be done with a computer, actually, this particular simulation. You cannot simulate with a computer because you have Avogadro many strands, if they're short enough. You have 10 to the 20 strands. But I'm going to show you how to do a multiplicative update with 10 to the 20 strands. And later on, I'm going to do an exponential blow-up that learns disjunctions over these strands using Winnow. That then would be, if you learn a 10 literal disjunction, would be you learn concepts 10 to the 20 and then power 10 again. It's mind-boggling what you can do with this very simple thing. Okay, so the fundamental thing is you start with a unit amount of random RNA strands. You can generate those. There's techniques for doing that. And then you have a loop and do a functional separation into the good RNA and the bad RNA. The good one is the one that attaches to the protein, to the sheet. The bad one that doesn't attach. And then you amplify the good RNA to the unit amount. Are you with me? So it's very boring multiplicative update. Very simple, no sex, okay? Okay, good. Now, there is a trick for duplicating RNA. So you have all the strands in your soup you can duplicate, okay? This was invented by Kerry Mullis. I usually show a slideshow on this, but over time, now more and more people know about this, so I stopped doing it. So it's invented by Kerry Mullis. He was in Lake Tahoe in a hot spring and on an LSD trip, and he supposedly invented this thing. It's the only result that I know of that was of that sort. So what you do is, and he actually was in the hot springs and he knew there's life forms here, and it gave him the idea, right? And so this life form had to duplicate, so maybe how do they do it? And then he asked that question. He extracted the life form and extracted a certain Taq polymerase that does it. Okay, so what you do is...
hot springs in Yellowstone?
Yes. In Yellowstone or in Lake Tahoe. You know, science, there's rumors. I don't know what's the truth. Okay.
I don't know which one he was bathing in, but the organism...
Okay, he got it from there. Okay, good. He was bathing supposedly. Okay, it doesn't matter. So you have your double-stranded... Everything is double-stranded. Then you heat it up, it becomes single-stranded. Okay? And then you have primers, that the ends of all of these guys are similar, are the same. And then you have short primers, they become doubled. So you have now a single strand, and at the end you have already doubled. And now you have a polymerase that walks around and doubles the single strand and doubles everything up. And now, essentially, you are back to a double-stranded DNA, and ideally, each DNA strand, all the DNA strands are multiplied by a factor of 2. Okay? That's ideally. But there's a hitch to that. You will see. Okay? So the main problem with this kind of research is it was very exciting back then. They wanted to do evolution. It's kind of like the genetic algorithm in computer scientists. In computer science, there's a lot of excitement. But it didn't quite go very far because it's very difficult to get smart functional separations that you can do at a high level. That's sort of the problem. And I've worked a lot on computational drug design that uses different tricks. But that's a separate talk. But now what I want to do is I want to formalize that multiplicative update, and I claim that using that tube you can run Bayes. Okay, good. So we name our RNA strands, the 10 to the 20. We just call them 1 to 10 to the 20. The naming is not important. It's just for the purpose of the description. Okay. Now, the SI... Bad, I don't see it. Oh, the SI is the share of the ith strand, the fraction. Okay? And of course, the share vector sums to 1. Right? It sums to 1. It's the share vector, and the total is 1. So this is a probability vector. So one of my tubes, the tube with my RNA strand is a probability vector. Okay, good. So now there's also a fitness factor. This is the amount of RNAi that attaches. So W5 might have 50% of attaching. This is a number between 0 and 1. That's this kind of number. So I call it the fraction of 1 unit of RNAi that is good, that attaches. Okay? So this is the fitness of RNA strand I for attaching to the protein. I'm trying to put some biology terms in there. Again, my cup, my tube, is a vector, the S vector. This is another vector, an attachment vector, the protein. Yeah? So you have two vectors. And now here it goes on. We don't know the vectors. We just named them. So we never go down and sequence the DNA and do something like that or know what the protein does. We just abstractly describe the vectors. It's kind of like kernelization in machine learning. We only rely on the dot products. And of course, every good science has an assumption. Otherwise, it's not good science. So the assumption here is that WI is the attachment vector is independent of the shares. It's a natural assumption if the concentrations are very small. Okay, good.
So the shares are the fraction.
The fraction,
of that
yes.
number.
So now I tell you what attaches. What attaches is, well, I have this much of RNA1. This is the amount that attaches. Okay, this amount of 2. So it's a dot product. Wow, now we have machine learning. We have dot products. Okay, the complement is what doesn't attach. It's of course the other part. The total is of course 1. Okay. Now, what's the amplification? Well, the good share of RNAi is this much. It's multiplied by a factor, and then afterwards it sums to 1. If you do a little calculation, you know that the factor is this much, and the final update that. In other words, SI is SI times its fitness over normalization. Now, if that is not a multiplicative update, okay, fine. So, you call SI the prior. WI call the probability of good given I, and the dot product good is probability of good. So I'm just using sort of high-level probability language. And then this update, bam, becomes posterior, which is the concentration of I given that you were good. It's the prior times data likelihood over normalization. So that's Bayes rule. So you do Bayes rule with 10 to the 20 variables. Okay? And you do it in your kitchen. You can do it anywhere by just pouring, right? That's how you do Bayes rule with 10 to the 20 variables. And you, in some sense, you don't even sequence, you know nothing, it's blind computation. Okay? So I'm going to show now, to start with this as my core, as an extremely boring multiplicative update. I will show you things cannot be done with this, and what you have to do to make it powerful.
So there's no mutations yet. You've got to fix.
No mutation. Boring. But I will introduce mutations in a moment. Okay, so now first I want to show you how these updates behave. So these are particular plots. Here are the concentrations. I have 3 species: green, blue, red. These are the concentrations: 0.6, 0.4, and a tiny amount of red. Okay? Now, this is time. So you multiply. What happens is, red fitness factor is 0.8. No, red is 0.9. Green is 0.8. Sorry for all the males in the audience. This is red, this is green, and this is red. And blue is 0.75. Okay? So you multiply by this factor and renormalize. Multiply by the factor, and each trial multiply by the factor and renormalize. Do you understand? It's a very simple update. This is the initial concentration, and this is the initial fitness. Everybody understand this plot? Now what happens is, initially you have this guy, which has a lower factor. Blue has a lower factor than green. Has a... right? So, of course, this guy wins out. But then this good guy that comes out of the dark, that had very low concentration, beats this one. And eventually this guy wins. So one guy wins. Okay? That's the main important thing that I want to make. Here's another one. 2 guys are very close to each other. Very little difference in the multiplicative factor makes brown get better than green. but eventually this guy, which has a higher factor of 0.9, beats them both. You can do all kinds of tricks with it. You can go to negative time. I cut out those slides, too many things. Okay, good. So the problem is the best are too good. Multiplicative updates, shares multiplied by fitness factor over normalization. But the blessing is you get fast. You can do it with 10 to the 20 variables because you can prove that the number of updates you need to bring it up is somehow related to the log of the number of things. And log of 10 to the 20 is not much. But the curse is the best wipe out others, and you have a loss of variety. Loss of diversity. I don't know what the right term is. Last time I talked to my housemate who is a soil scientist. He studied ecology. Which word should I use? Diversity or variety? We got a huge argument, and he mentioned evenness and all kinds of other things. It's simple. And we actually agreed on diversity as the word. Okay. So the simple view of evolution is you have inheritance, you have mutation, and you select for the fittest with the multiplicative update. Okay, now the new view is, and this is what I'm going to talk about here, is that the multiplicative updates are extremely brittle. When you put things into the exponents, things goes bad very quickly, very dangerous. So a mechanism is needed to ameliorate the curse of the multiplicative update. It's going to be short-term versus long-term. The short-term is going to be multiplicative, and the long-term is always something else. And machine learning and nature uses various methods. I will show you the ones we found, and you can ooh and ah then and see, oh, does nature do this as well? You'll see. Okay, good. So let me show another tube experiment. You take a tube, and you put in 100 different bacteria in there on a nutrient solution. And you just let it sit. How many survive? You wait, there's competition. And typically, and people, I designed these experiments and then I found it in papers. 3 survive, 1 in the muck on the bottom typically, 1 on the side, and 1 on the surface. So they found 3 niches. 1 in the muck, 1 on the side that was still light, but it's still oxygen destarved, and on the top you have oxygen. It's order of 3, of course, right? So a small number survives. Now here comes the fundamental deep question. You do the same experiment, but you agitate the tube. You put it on a magnetic stirrer. I do that a lot when I grow spirulina and stuff like that. So you have a magnetic stirrer, and you put it on, and then you do the same experiment. And now how many survive? 1. Why? Because you have only 1 niche.
No, you often get many, but that's...
No, well, yes. First order approximation. Okay, it gets very subtle. Because it turns out that the genome is part of the environment, and it's very messy. So you don't get total mixing, in some sense. Okay, good. So I believe that this is actually one of the main problems of the human endeavor. It's not necessarily global warming. What happens is global warming is one of them. We are connecting the world with roads. Roads could be tools that you invented. It could be an iPhone. It could be all kinds of things. I call them roads, in quote. And they cause mixing, and therefore we have a loss of variety. So this is as a world citizen, I'm very much concerned with this. I believe the human endeavor enhances right now the curse of the multiplicative update, and we have global die-out. Why is this bad? You figure it out. Okay? I could talk about that. Okay, let's go on. So now I'm going to show you a first key problem that cannot be solved by the multiplicative update alone. Okay? And they have to have a fix, and then I show you how nature fixes it. And it's an amazing little fix. Okay, so here's my tube. I drew it as a square. I apologize. And you have some concentration of 3 strands in there. You don't know what they are. They're not separated like this. It's a soup. And the task is, how do you make more of the soup with a multiplicative update? I claim you cannot. In particular, you cannot do it with PCR. If you apply PCR, these RNA strands are folded ever so slightly. They hug the Taq polymerases. C might have a survival factor of 1.91, and this one would be 1.8, and this one 1.97. So if you apply the multiplicative update many times, PCR, in many states, you can do this automated now, robots can do it, very, very fast. What happens is, one guy will win. So this is a task that the multiplicative update cannot do. You cannot, competition causes die out, diversity is lost. So how do you, does anybody know how to fix this problem with RNA technology? Is there any RNA people here, DNA people? I'm only a layman.
Reverse transcriptase.
Yeah, something like that. So what you do is, you add stickers to the end of the RNA strands, and make a long, and they stick together, you can do that, and you make a long strand. And now, you have this long strand, and then you can put a special primer at the beginning, at the end, and you fish out one of those long strands, or you pick, and on the long strands, roughly the right amount of A, B, and C are on these strands. There's an error, of course, because you're random sampling. And there's a big error, it's not precise, but the point is, once you have the long strand, the error doesn't blow up. Okay, why? Because you have a primer at the beginning, at the end, and now you amplify, you can set it up so always the full strand is copied. The full strand is copied. You can do that using primers at the beginning, at the end.
You make one long strand.
Yes.
You mean that the randomness. I mean,
you take your soup, you add these sticky ends, they attach to the end, and then they make long chains.
They're not A and C specific.
No, they're not A and C specific.
Okay.
Yes. They're not A and C specific, they correct, yes, and there's an error, of course, but it doesn't blow up. And then you do PCR on this, and then you chop into constituents. And so the long strands function as chromosomes, and free-floating genes in the nucleus would compete, you would get loss of diversity. And here's the picture. So what happens is, if you have them A and B in parallel, apply PCR, competition, one guy wins. If they are linked together here, and you apply PCR, then actually you compete for the best variant of A and B, and it forces a cooperation. So it's like this. There's a handshake, and actually you can see it's cooperative in some sense. It looks like that, but then there's a tie knot. You're forced in this together. So I would be competing, Wouter and I will now compete with other variants, Leslie and Christos, who also may be a pair. We compete, and now we are not competing against each other, we are competing against other pairs. And this is a fundamentally deep thing, and this is how nature solved this problem, using this kind of coupling. There's all kinds of tricks in the cell that make sure that everybody gets copied and it's fair.
There's a question.
Yes?
So when you have all these, you put these sticky things in there, right?
Yes.
So in your tube you're going to have a lot of long strands.
Yes.
Some of them will have 45% A.
Yes.
Some of them will have 50.
Yes.
So
You pick a random strand.
Oh, so these don't compete against.
You pick a random strand. You can thin out onto petri dishes or whatever and figure out, is there a strand there? And then if you have a random of the right long size, you use that.
So you're able to pick one at random.
Yes.
Put it outside.
Yes, that becomes your chromosome, and then you apply PCR to that. Okay? Good. So we cooperate. Actually, I went to the sex store to get handcuffs, but I forgot them in San Diego. Okay. So there's many questions already about in vitro selection. Okay, which is a bare bones evolutionary process. Okay? So what updates represent an iteration of in vitro selection? What updates are possible with blind computation? And the crucial thing that we always are forced to do when we have concentration is, when we have this kind of thing, we have to update concentrations, non-negative weights, and we somehow, there's not many updates left, because you have to update the concentrations.
So at this point there's no selection, they're all being amplified together.
And they amplify together, but the selection is based on the fitness factor. There is selection. Selection I have already. I haven't talked about mutation yet.
You selected first and then you're amplifying.
Okay, in this particular experiment, in the earlier one when I did Bayes rule, I had selection. In this particular experiment, what I'm saying is, if you want to do this thing, you get a long strand, and then you just duplicate and get more of the same soup. Okay? There's no selection here. Yes, in this particular thing. I'm going to do other ones. You'll see.
Sorry,
Yes.
so after you do this tensor, whatever, to put together the two, how does the fitness change?
Well, it depends on both. The fitness depends on both, and now you are going to select for fitness, select for good combinations of A and B. And I call that A and B sort of cooperate. They have to cooperate. The genes have to, our genes get our bodies out there and do all of these things, make love and do all of these things, and work and come to lectures so that we get more offspring. It's a pretty roundabout way. It's kind of unbelievable, but that's what is believed to be biology. Okay? The genes drive it. Now I'll give you another example. Another example where the multiplicative update by itself cannot solve. And this was solved by Winnow, which is a little bit of a contradiction. So, but I will bring it out in a moment. So you have the same thing with proteins as before, but you have different proteins. In one trial you have protein 1, you protein 2, you have a bunch of proteins, and you want to find a soup that attaches to all of them. So in some sense you want to find a union that does the job. And you want to define it in a decoupled way. Okay, so each time you do your thing, tip P1 in there, wash off, and amplify. Pick P2 in there, wash off, and so forth. Can you figure out how to attach to all of them with blind computation? You will see a multiplicative update by itself has a problem.
Blind, you mean without knowing the WIs?
Without knowing the W, yes. Without knowing anything about what's in the soup. Okay? Okay, so look here. Assume there's two guys in there that do the job. K equals 2. You know, there's one that attaches well to these two, high factors, and one to these. You see? And ideally what you want is you want to have this tube with where everybody has concentration 10 to the minus 20 and arrive at this tube, because that does the job. How do you do that with blind computation? Winnow figured this out. Okay? That's Nick Littlestone.
You have to explain this a little more.
Yes. Calm down. Okay. So now, the goal is you start with a tube where everybody has frequency 10 to the 20 and you want to arrive at this. So here's...
Specific two, or you want to arrive at all possible pairs?
No. You don't know what the two are. You assume you have a tube and assume two can do the job. Can you find them? Can you do the job? Can you raise their concentrations together? You see, in a decoupled fashion, because they are not coupled on one gene, W1 and W2. This is free-floating first RNA strand, free-floating second RNA strand. So I go against what I did with a handshake here. But still there's a fix. It's totally fascinating. Okay, so you do this. Okay, how do you do this? Well, if you overtrain with P1 and P2, if you overtrain with these two examples, then the first RNA strand will blow up and will dominate. You have a multiplicative update, huge factor differences, and this guy will dominate. If you overtrain with this, you have then the second one will dominate. And this is a problem. It is highly unstable. Okay? Here's how Winnow fixed it. Okay, you want to do blind computation. So here's the abstract problem for the machine learners. Your instances are bit vectors. Your weight vector is that you're looking for is a disjunction, 1 over k and k precision. And you want to look for something that this is sort of expresses the disjunction, and you want to use this thresholding to figure out whether you're in or out. So this is sort of the machine learning thing. How does the update work? The update does the following. This is the good side, the good RNA. If that is bigger than a certain threshold, then what you do is you do not update.
Why is W a function of T?
Because each time a different protein sheet is tipped in. I called it WT.
I thought they there were K different.
There's K different things that do the job.
Oh, I see. Got it. And there are.
Yeah?
Sorry.
Yeah? Okay, good. So if a lot of it attaches, then you don't update.
Wait, sorry, you have WT1 and WT2. You have two different sets of RNAs, or that?
Okay, there's two things going on. There's the weight vector, in which case where you have two strands that you want to select for. And then you have tasks. And the two strands cover the task in some sense. In the sense that the first has a high factor on some of them. The second one has a high factor on some of the proteins. Together they cover all of them. It's a disjunction. That's the idea.
Together they cover all of them.
They cover all proteins. Look at this picture. This picture here. There's a red factor, a high factor on all 5 cases. They cover all of them. That's the point I want to get across. So they cover all of the sheets, but you don't know what the cover is and so forth, and you still want to do it. And the way you do it is you do this kind of update.
Should imagine that there are many rows.
Yes.
Different color, and
Yes.
basically 2 colors cover everything.
2 colors cover. It's a set cover problem
Right.
for the computer scientist. And so what you do in this, you want to do blind computation. So you do a thresholding, bam, and if the good side is large,
I'm sorry, you've gone now from WT1 to WT2,
Yeah.
back to just WT. So what is the
WT is
relation of
vector.
WT to the...
at each trial, I get my current vector of concentrations. I see what attaches, and this is the protein that I tip in, right? I measure the dot product, and if it's bigger than the threshold, if a lot of it attaches, then I don't do my update. And I will show you in a moment why I don't do that. I just don't do PCR. If, however, not enough attaches, if you're below the threshold, then you do a multiplicative update. And you can show this is information theoretically optimal. The number of updates of this type you need is k times log n over k, which is the number of bits to encode the disjunction. We do proofs, we do everything. We know that Nick Littlestone did this. Yes?
Are you going to tell us?
No. That's 30 years old. Okay, we'll get to the new stuff. Okay, so now, so this is the first trick in...
Was this Winnow?
Yes. It's a version of Winnow with all the examples being positive. I did normalized Winnow for some reason, which is a very...
I knew Winnow. Okay.
Okay, good. So ML1 is you prevent overtraining using the conservative update. That's the first trick. So but it assumes that you can measure this concentration. So if you call this still blind, it's a dot product, okay, then you're fine. The normalized Winnow can be implemented with blind computation. The algorithm uses n weights, 1 per RNA strand, a concentration per RNA strand. I didn't go via the coupling trick. I didn't make up one RNA strand per disjunction. Then I would have 10, assume I have a 10 literal disjunction. I would have 10 to the 20 to the 10 different things. That's too much. This is too many, combinatorial blow up, but I still can learn this many concepts efficiently. It's mind-boggling trick. Okay.
Can you say, what is N?
N is the number of strands in the soup. K is the size of disjunction.
Is what we are trying to learn is basically the things that we know.
I had K equals 2.
Out.
I had K equals 2 and N was 10 to the 20.
Okay.
Okay, good. Now, so you can do this. So now I'm going to do other tricks. Next trick, a cap weight. And I actually learned this from my class with Barry Sonero and Dan Friedman.
The K you have to input a priori, or
You need to know K to tune it. But we are working on this non-stop, I tell me, for the last 30 years, work on these non-steps to try to get rid of these tuning parameters. We got rid of one of them recently using dropout. It's a long story. Separate talk.
and K has to be less than the number of tubes, just logically,
And K has to be the number of literals that you're going for.
which is less than the number of tubes that you have potentially to select on.
Yeah, sure.
How do you do the thresholding biologically?
Biologically, what you do is you would, and I talk to DNA guys, you would look at your tube that you washed, the stuff that you washed up, and you send some kind of radioactive, you need grad students to do this, you can't do this. Send some kind of radioactive light through it, and you measure.
The students are going to wait for you outside.
So the next thing is going to cap weights. Okay, I need to go on. Sorry. Huh?
You don't need K-tubes with this. You only need two, I think.
I'm not doing. I have one tube that I train with, but I tip in different sheets.
Let him finish, I want to hear you.
Okay. So now, so here's capping from the top. So this is the predator idea, the super predator idea. If you have a lion species in the Serengeti, it's specialized. It has to invest into what it learns to hunt. It takes a lot of time to learn how to hunt buffalo, Cape buffalo. Very, very nasty animal. Has highly defensive. It's a match. So they have to learn how to match to hunt them. So essentially, they have to they specialize on the most frequent prey. So in other words, they eat off the top of the bar. Okay? And I designed caching strategies using this and so forth, and eventually we found an amazing application. So here's your simplex. You update with multiplicative weights, but then if you cap, let's say you cap with a half, then it turns out you have a smaller polytope, and this has exponentially many corners. What you do is when you cap is you do the multiplicative update, and the weights that's outside, you take it away and distribute it proportionally. This is sort of technically what happens. Motivated with relative, and everything is always motivated with relative entropy regularization. So what happens is, these corners of the simplex correspond to sets of size 2. Here, tetrahedron, 4-dimensional simplex, represented as a 3-dimensional tetrahedron, then this is an octahedron. Each corner of the 8 corners corresponds to a set of size 2. And now you apply multiplicative update in this context. You do a multiplicative update, you cap. And now you learn exponentially many things. You run into the corner of that simplex, and you have exponentially many corners, and you have an exponential speed up on top of the 10 to the 20, but I do not know how to do it with in vitro selection. I have all kinds of theories, but I'm not an RNA
Sorry,
technician.
exponentially, because you have exponentially many corners.
Yeah.
But do you select a convex combination of these?
Yes, you're working with a convex combination, which is a mixture.
Right.
You do a multiplicative update, you go outside, you project back in, use an entropy projection, which you can implement if you can do capping. Very, very beautiful trick. It's a similar trick as Winnow, slightly different things.
The corners are then best, or I guess...
You go for the best set of experts in this context. Okay? Okay, good. So now, here is what we did for a very long time. I did it with many students, Mark Herbster, and lots recently with Wilder and lots of people. Okay? So you have 10 to the 20 different strains, and after a while, you have loss of diversity. Some selfish strain manages to dominate without doing the job. In vitro selection people know this all the time. They create an initial soup, and they do the individual selection experiment, and then the soup gets exhausted. Some bastard gene took over, it's totally useless, and you can't get rid of it. So what they do is they pour in a little bit of the initial soup. And we did this with machine learning. So you mix in a little bit in soup for enrichment. You can do sloppy PCR, you can do many tricks. So we discovered this, for instance, we apply these online algorithms in many contexts. For instance, disk spin down problem. You have you want to figure out when to spin down your disk, and you want to adapt to the behavior of the user. If you have a lot, these are the idle times. If you know the idle times are going to be short, then you should have a long timeout. You should wait it out, because it takes a long time to spin it back up, right? A lot of energy loss. And if you have long timeouts, then you should spin down immediately. And if you do one of those multiplicative updates, which is natural in this context, you make up some disk spin down times in the right range. You update using e to the minus usage of the energy, and then what happens after this? All the species, the long species died out. The long timeout died out. So you're in trouble. So you need to have a fix. And the fix, because you want to now favor the other ones. So you want to do the multiplicative update to home into the appropriate disk spin down time. But then you also want to have long-term changes. What if the Earth gets hit by a meteorite? How do you solve those problems? And nature solved this, and we solved it in machine learning, and the interconnection is mind-bogglingly interesting. So what you do is, you can take your multiplicative update, instead of projecting now, what you do is you take alpha is 1%, 99% of the weight vector you just got, and you mix in 1% of the uniform distribution. Yeah? That's all. Now, this makes this thing robust against change. Okay? And maybe nature uses mutation for that purpose. Okay? This is one of my weakest statements. Please don't hold me to that.
Uniform distribution,
Yeah.
not 10 to the 20,
Yeah.
you don't have 10 to the 20 in your soup.
Yes. Yeah, in nature you don't have 10 to the 20, whatever the number of genes is. It's your n, the n0 or whatever. Okay, so, but there's an amazing, what we found out, this is kind of neat. You can talk about this forever, you can model it, you can have a Bayesian interpretation over exponentially many experts and do all kinds of things and explain it, but there's a fancier
Manfred, I have a
one.
question.
Yeah.
Do you like this stuff?
What?
How much do you like this stuff?
It's very subtle, huh? Okay.
A better analog to mutation might be immigration.
Okay.
Cases that might work. It's hard to justify some mutation, but I think you
Yes,
have a better analog.
some kind of process that mixes, that keeps the lower bound, in the weights. So now here comes a key thing. So imagine you have 10,000 genes, and then for a while you have nature where A dominates, has a high fitness factor, then D dominates, has a high factor, then G, then A again, then G, then D, then A. You see, I don't tell the algorithms this. I just run one of those algorithms. So this is I measure the loss of the algorithm. So I always let it vote with a particular gene and then measure the loss. Okay? Good. It's very robust. You can do all kinds of loss functions to do this. So this is how the total loss over time grows of the best partition. God knows what the best partition is. That's what I'm after. This is the loss of a partition of an arbitrary expert. It goes up much faster. Now, here's the guy that does multiplicative updates. Multiplicative updates. It burns A into its weight space. It cannot learn D at all. It has the bad rate. Don't know why this is flat.
Eventually it learns the...
It got it. It got it at this point. Yeah, it was long enough. Or it took a long time. If you run this longer and it wipes out completely, it would go all the way straight. And then it, again, here it remembers it immediately when it goes back to because it learned this other one now. I see. Good. Okay. Now here is the one that. This is the one that mixes in a little bit uniform distribution. It has the same size bump each time, the black one. It can recover rather quickly. Now there's one update which has a long-term memory, and that's this update. Instead of mixing a little bit of the uniform distribution, what you mix in is alpha times the past average distribution. So you keep two n weights, and you mix in a little bit of the past average. There's another scheme that I will show you in a moment.
Cumulative average?
Yes, cumulative average weight. Okay? So you can do that with pouring again. You soup, you pour and pour and pour, and you can do this. You need 2 cups. That's all you need, 2 cups. And a little calculator, okay? So you do this, and that update has the property that when you come back to A again, you only have a small bump. So it learns A, and then you come back to A, and it's a small bump. Here's A again, and it's a small bump.
This is somehow like the immune system is supposed to work.
Exactly. It's all over nature. There's a long-term memory. If you not learn songs, if you have learned a similar song before, you can learn it again. Okay? With genes, if you have a species that was resistant to some kind of sickness, if later on the same sickness shows up, you remember it fast. The combination, this one line, in multiplicative updates we talk about a lot. I show about tricks, right? Capping from the top, lower bounding from the bottom, all kinds of tricks. If you add this one line, the algorithm does amazingly different things. It's just mind-boggling. It does this thing, it has a long-term memory, and I actually won a prize with this for my student. It's the only money that I earned honestly. But we won the prize, and then we put it on harder problems that so my money is totally safe. I doubled the amount and I put it in a harder problem, so it was fine. Okay, so the punchline again is, whenever you rediscover something, the bump is small.
And best partition means what?
Best partition is, in hindsight, God knows what the environment is. It can optimize locally for the best gene, and would be the best partition in hindsight. Okay, we learn well against the partition in hindsight with a very simple update that has 2 lines of code. Now here's what happens in weight space. The good guy is pulled up fast. And now, when A comes again, it's pulled up faster. This is all exponential weights. This pull-up is faster. Or the first time green is pulled up, it's this, and here it's pulled up faster. So what happens is you have a long-term memory that brings out things that were good in the past are brought up faster by changing this 1 line of code. There's another one, how you do it with cups. So here, so you do a multiplicative update, and then you do a little soup exchange. You will see. So here's the multiplicative update. Selection plus PCR. Here you can put in a live model or whatever you want. I purposely stayed very simple. Selection plus PCR, right? So it's very clear there's no sex involved. Okay, then you have another, you keep 2 cups. Initially you partition it into 2. Here's your other one. And this one you just pass through. And now what is this step? You just pour epsilon here and epsilon here. Okay? Then you do again your multiplicative update on here. You tip in your sheet and you do this whole PCR thing. And then you go, right? And then you do again PCR over here and you go, and if you do this thing without knowing anything, it has a long-term memory. And we have an interpretation of this. We proved reasonably. This update.
Epsilon stays the same?
Huh?
Epsilon stays the same?
We keep it fixed, but there's tuning problems, working on those. But the point is, I worked with some of the best machine learners, and I drove them totally crazy because we didn't have a Bayesian interpretation. You see, how do you get a Bayesian interpretation? The Bayesian interpretation would be you want to have a prior on all possible partition experts, and then do an update, blah. You see? And if you choose the prior right, blah, it works. So if you lower bound the weights, or you mix in a little bit of the uniform, very easy to get a Bayesian interpretation. And for this one, for one mixed in the past average, or the one that I showed with a cross pouring, very hard to get a Bayesian interpretation, drove us crazy. But then Wouter and Mitya Adamski, they figured it out, and we wrote a paper, Putting Bayes to Sleep, where you can show that if you have exponential many partition experts, and some experts are asleep in the sense that they don't vote, they don't participate in the PCR. They just sit outside and vote with what everybody else does. They abstain from voting. And if you use this additional abstaining trick, which is also apparent in this collapsed version where this whole population in some sense abstains. We don't fully understand all the connections, but we have proofs that this thing works. In an upshot, information theoretically, what do we do? Imagine you have a million genes, and you want to learn, you're shifting. The environment changed, catastrophic events. First century, gene 5 is good. Second century, gene 20 is good. Then for 100th century, nothing happens. And then another gene is good. And you always shift around between a small number of genes. We show that these kind of schemes have the following information theoretic property. You pay log of a million to first learn, log of a million per gene the first time you use it. The second time you have to switch to it, you only pay log of 10. So it's very, very powerful.
10 being?
10 being the size of the small pool that you shift between.
Okay.
The first time you pay a lot, it's information theoretical optimal. It's related, what appears in the bound is the number of bits to encode this. And of course, if you have repeats, your encoding gets better. Okay, so here, how is long-term memory realized in nature? Of course, a lot of you have thought about this. Junk DNA, sex, of course. You know, we have already a paper in progress. It's called, we have Putting Bayes to Sleep. In the next paper, we already have the title, Bayes and Sex. But we have technical problems.
That's evolutionary listing.
So we did. I showed you the conservative update, upper bounding the weights, lower bounding the weights, mixing, past average, sleeping. Nature. I showed you a few of them. Of course, there's many, many more. I don't know how to prove things about sex, blah blah blah. But here's a bunch of other ones. Okay. So multiplicative updates converge quickly, but they wipe out diversity. Changing conditions require reuse of previously learned knowledge, and the diversity is a requirement of success. And again, the danger is that what humans are doing is we are eliminating diversity because of the mixing that we are doing, and we do other tricks in machine learning. Okay. Now I show you one more open problems. Okay. Remember, I wrote out SI was the old SI times e to the minus eta loss. A generalization is gradient of a loss. By the way, Winnow, the conservative update of Winnow can be explained using a threshold loss function, which explains the conservativeness. But you have to use the gradients of the losses. Okay, so you have your multiplicative update. In matrix case, you cannot multiply two matrices and preserve the properties. What are the properties of matrix that we're looking at? Before we had S being a share vector, a probability vector. Now it becomes a density matrix, which is a matrix where the eigenvalues are a probability vector, but you also have an eigensystem. And if you want to do the multiplicative update there, you first have to take a matrix log of the matrix, subtract the gradient of the loss, and then matrix exponentially divide by the trace. We have bounds for this. There's a whole Bayesian probability calculus based on this. It goes on and on and on. But I do not know whether nature uses this update. Whether there's an implementation of it, like with in vitro selection or something like that, a rudimentary experiment. It's kind of a mind-boggling open problem.
Actually,
This one, you need to, this had many convexity classes collapsed recently based on this update and so forth. They had major influence in complexity theory. A lot of approximation algorithms are based on this matrix version of the Hedge algorithm. Okay, and the key thing is, what motivates this thing is the quantum relative entropy as a regularizer instead of the entropy. And that goes back to Van Neumann. Okay, that's all. Any more questions?
Are you aware of the work in the replicator equation? On
Yes.
Coexistence of parabolic replicators. So if you have a parabolic replicator, the rate of change of x equals x to the power of something less than 1.
I know the replicator equation. It has to do with the multiplicative update.
But there's a variant like von Kodrovsky and Rajnath Mari, which looks at the coexistence of replicators if you have an exponent of less than 1.
Yeah.
So xt plus 1 equals x to the power of something less than 1.
Yeah.
Then you have a similar result to what you're doing with this artificial threshold thing.
Okay, good. I would love to see that.
Coexistence of multiple replicators with different replication rates.
Aha, good. Yes. So this is kind of the interesting thing. Our updates are so easy, you can analyze them. I really got ideas from nature, believe it or not, even though I only read at a mad scientist level. I'm not a biologist. But the connection is kind of mind-boggling.
Do you know Harper's work?
No, please talk to me afterwards. Actually, I'm promiscuous in science. I work with anybody. Okay, so please talk to me. Actually, you can test. I have tests for testing whether there is a long-term memory in any system. You can do a psychological test for it. You can do a biological test for it. I don't know yet how to quantify quite how much long-term memory there is, but with experiment you can test it. And this is kind of, I think, the next thing that has to be done. And then once you test it, maybe you can knock out sex and see whether sex is responsible. Maybe you can knock out various features of biology. But we know it exists, it's useful, because we work on the same problem. We know we have proofs that it works, and now we should test for it and systematically dissect.
You mean long-term memory, the existence of long-term memory?
The phenomenon that when you have a huge number of genes, and you have catastrophic events that are long enough, and you shift, and you recover faster if you had that resistance long time in the past. Yeah? Very, very, very deep question, and that will really show experimentally that this happens in nature, and then I can go in and modify the nature's toolbox and see which one does this. Okay, any biologist who wants to do experiments, please talk to me afterwards. Yes?
I'm very interested in the notion of evolutionary processes having long-term memory.
Yes.
The kind of memory that you were showing here is a sort of rote learning memory, right?
Mm-hmm.
You take a snapshot of something that was useful in the past and you keep it in case it's useful in the future.
Yes.
Other kinds of memory are sort of structural memory, which have the capability in principle of doing some sort of generalization. So
Yes.
you're faster to adapt to something because you've seen things that were like it in the past,
Right.
but not exactly the same.
But we're doing this as well, because what happens is everything in machine learning is weight vectors and there's predictions and so forth. It's exactly that kind of thing. If you are similar to something in the past, this weight will come up because it has smaller losses, a smaller gradient of losses, and it will be learned faster also. That we do, yes.
So that's like weighting over a feature space. So it's similar in feature space.
It's similar it would be the relative entropy between, you need a certain concentration vector to do a job, and if the relative entropy between the one that you want and the one that you had is small, then you would learn faster. It gets technically very fast. Apologies to the biologists, we look at very, very simple setups here, but we found some tricks that look intriguing. I mean, the connection is not far-fetched, I think. Yes?
Sorry, I've already asked.
Okay, sorry.
I mean, if your task was to find the best pair of RNAs
Yes.
whose binding was optimal, so
Yes.
there's some pairwise
Yes.
thing, is that fundamentally different for you?
Okay, if you
Is that
look,
a mistake?
when you want to find, let's say you want to find a combination of 10, right? So you have two choices of what to do. You can make up little chromosomes with 10 genes each, and then have the species compete based on the computation of 10 and do the multiplicative update. We do that sometimes, but we call it expanded hedge. But what we do is tricks to let us keep on working with the same number of genes. But we do a mechanism on top, and we provably learn combinations of 10.
I guess I'm asking about if there's a pairwise interaction between everything in your soup, and you want to find the pair, so potentially n squared. So you
Yes.
want to find some subset of the n squared for which best bind. Is that within your lexicon?
Bipartite the general graphs.
It is within the lexicon. I haven't analyzed it. You can do all kinds of things in practice. If you have a weighted combination that works really well, freeze it and throw it in your gene pool. These kind of whenever you do entropic regularization, you can do amazing things. What you can do is and this is really a portion of something of the simplex. You can go, if something doesn't work well, it can go to an extremely low concentration. It can go back to 10 to the 20. As long as it sticks around, as long as it lingers around, it's fine. And then it can be picked up again. So you can really have a long-term memory of size 10 to the 20. While running and doing optimal things, and always bring it back up. It's mind-boggling, and it's a property of the simplex.